welcome to the Talking Crop podcast. I'm Kathryn Seebruck, a commercial agriculture educator with the University of Illinois Extension, serving Daviess-Stephenson and Winnebago counties. Talking Crop is a seasonal crop production podcast with episodes occurring every other week during the growing season between the months of May and September. In today's episode, I talk with Dr. Kathryn Siebruck, a postdoctoral research associate with the University of Illinois Crop Physiology Lab. We had a really great discussion where we first covered findings in his recently published article on continuous corn residue management and its effect on the soil microbiome. Then we talked about how the growing season is going and what to look out for as we head into June. We ended by discussing soil health testing, as well as the upcoming Crop Physiology Field Day in August. So please be sure to evaluate the Talking Crop podcast at the link in the video description. It's very short, and it just helps to give me an idea of what you, the audience, want to hear about and what guest speakers you want to hear from. I've also included a link to the Crop Physiology website, which you can visit to learn more about the Crop Physiology Lab, what research they do, some of their research findings, and it's also the place to go to get more information on their field day as it becomes available. And on the next episode of Talking Crop, I'll be discussing soil biology, plant-soil interactions, and how cover crops influence the soil microbial community with Emily Hansen, another commercial agriculture educator with University of Illinois Extension. That episode will air on Wednesday, June 12th. So without further ado, please enjoy this episode of Talking Crop, talking continuous corn, residue management, and more with Dr. Kathryn Seebruck. So Connor, thank you so much for being here today on the Talking Crop podcast.
Thanks for having me. Looking forward to it.
Yeah, absolutely. So before we get started with our conversation, I wanted to have you give the audience some brief information on the Crop Physiology Lab. So I always learn a ton by listening to whether it's your talks or your colleagues in your lab giving the presentations at different events. The stuff you do is really, really interesting. And so I was hoping that you could give the audience a little bit of background on what you do and what you guys work on.
Yeah, so specifically my role, I am currently a postdoctoral research associate, which means I've finished my grad work, but now I'm what I sometimes claim that as professor in training. So that postdoc role, trying to teach you things that it takes to be a faculty member. So I work with Dr. Fred Bilo. Many people know who that is. And together we run the Crop Physiology Lab here on campus. In short, what we focus on is yields and high yields, and how do we achieve that through agronomic management practices. We kind of look at everything from management perspective, right? Fertilizer inputs, look at practices like row spacings, a little bit of till versus no-till work. And then we also do other inputs specific to my area, biologicals, biostimulants, all about how they interact together to get a yield response, where the physiology comes in play. And I think this is the most important part about why we do what we do is, for the most part, anyone can kind of put a trial out and compare yield A to yield B. But what you really want to be able to do is say why. Why was treatment A different yielding than treatment B? And that's where the physiology comes in. And what we do is, when we have these practices, actually be able to tell the farmer, this is what's happening in your field to then cause that yield response we're looking at. So that's kind of a quick summary of the Crop Physiology Lab. We've got 5 master's students and a PhD student currently, so a pretty strong program moving into the 2024 season.
Awesome. Yeah, like I said, I always learn so much about a lot of things from your talks and your presentations at different events like field days and such. And I know that you recently, you were the main author on an article, and it's titled Long-Term Continuous Maize Impacts on the Soil Microbiome and Implications for Residue Management. So it's a word-packed title. So could you maybe break that down a little bit and talk about the background of this study, kind of the premise behind, what maybe was the problem that you guys were looking at and trying to find more information about?
Yep. So based on the title, right? So maize, because anytime we publish, we go with the international terminology, but we're talking corn here, continuous corn. And there is ways to make continuous corn yield, right? We know that corn soy rotation is the dominant rotation in the corn belt here in the US. And we do this because there's often a yield penalty of corn on corn. A couple reasons for this. You do the same crop year over year, you can have pathogen buildup. So then you've got pathogens that make your disease pressure higher. We've got a lot of residue that can tie up nitrogen. So if you look at a lot of the agronomy handbook recommendations, if you're corn on corn, you're typically going to need a little more nitrogen. And those are some of the big factors that can then limit yield. So some of the questions, though, why do we do corn on corn then? Corn generally, in most years, is more profitable than soybean. You get more bushels, and while they're lower cost per bushel, there's an economic play there. And so if you can manage corn on corn, maybe a 2-year, 3-year, or in this case, a long-term rotation, with management, you can make it economical as well. So that's why maybe some growers are interested in corn on corn versus just sticking to a corn soy rotation. And especially, you and I are both from Northern Illinois. There's a lot more continuous corn in the northern part of the state as well. So that's kind of where it started. If you are doing corn on corn, what practices might you need in that system to help you minimize that penalty, to keep that profitability up if you are not doing a standard corn soy rotation?
And so I want to talk first specifically about kind of the latter part of the title. So the implications for residue management. So you talked about with the continuous corn, that yield penalty. I know a lot of it comes from those residues left behind, and this comes comes to fruition truly with a lot more folks doing minimal tillage to no tillage. So can you talk about some of the treatments that you guys were looking at regarding residue management and what you found?
Yes, and I think I'm going to go back to your point exactly with the increasing tillage component. The number one recommended practice for continuous corn would be tillage. Work those residues in, try and get them to degrade. But there's a lot of pressure and interest in looking at reduced tillage systems. So in this work specifically, I'll give an example. It is a long-term site. I think we were on 17th or 19th year continuous corn when we started these the two sister sites in the study. And that's a lot of continuous corn, long time, compared to corn soy rotations. But we took it to no-till, which would not be a typical recommendation, but it's kind of a worst case scenario. Or if you are interested in no-till corn on corn, what options beyond tillage might you have to manage the residues? So when I think of research, there's, half the projects I do, I would love to say what I do today, a farmer could do tomorrow. The other half of the projects for what I'm doing today is to try and make sure we have the answers that will be needed in that 5 to 10 year mark. So this project, I would say, is more in that bin. So no-till continuous corn. If you're not able to till or in a reduced till system, what other options do you have? We looked at sizing the residue with the combine. So that starts with the previous fall's harvest. Looking at a little bit of ammonium sulfate, and maybe we'll get into that a little bit specific about why we do that. And then we started looking at a biological component as well. I think the residue degrading biologicals is a big area with a lot of unknowns. So we wanted to try and get some answers to that. So I would say those are 3 alternative practices to tillage that we wanted to try in this system to see if we could get a yield bump to those.
Okay, yeah, that's really interesting. So basically, you were looking at 3 kind of residue treatments, so to speak. And so, did you look at them, the effects of those individually, and then how they are when combined with one another?
Exactly. We looked at every possible combination on their own or together. The really exciting thing about the study was in both years, 2 different sites, more or less anything we did was in the right direction. So the no management was always the lowest yielding, and then any sort of the management that we did was higher. But it was very clear that if you put combinations of 2 of those practices together or 3 of those practices together, it was always better than any one on their own. Then you start playing with the economics, right? So thinking about sizing the residues with the combine. That's a pretty big investment to buy the different knife rollers for your combine for every row, but they last a long time. So that's a one-time investment that then can maybe bring back profitability over time every time you use it. Whereas the ammonium sulfate and the biologicals, you pay for that every year. So what's the price of fertilizer? The price of the input might vary a little bit with that. So that's another important thing to always remember when you're picking practices, the cost value to it in the background. In general, though, we liked sizing the residues, adding a little fall ammonium sulfate, and then putting a biological on top of that. Sizing the residue exposes the surface area so that the microbes can get to it faster. So just kind
Mhm.
of making it easier to eat, like cutting up your steak before you chew it and not just grabbing the whole thing and taking a bite out of it, right? Is the way to think of it there. Ammonium sulfate then is like seasoning the steak a little bit, right? A little salt or something, make it a little tastier for the microbes to go after. Corn residue has a lot of carbon in it, and you need a little
Mhm.
nitrogen, a little sulfur to help it break down faster. So that's why we did the ammonium sulfate. So you've exposed it with the chopping, make it a little more easier to chew on for the microbes, season a little bit with the ammonium sulfate so they can break it down even faster. And then the biological is just to give it a head start on top of that. So that's why those three practices were selected, as opposed to just tillage as maybe a standard control that we would normally think of.
Right. Yep, that makes a lot of sense. And then you talked about how combining these practices ended up with, the biggest impacts on reducing that yield penalty. Looking at the practices individually, though, was there one that ever like stood out amongst the rest, or was that one of those where it just kind of depended on the conditions?
Yep. So definitely the sizing of the residues was probably our biggest. It was our biggest yield benefit if you were to pick one,
Okay.
and that's at harvest on the combine. We're not going. The sizing knife roller would be what we had on the combine head. And then very close to that though was the alternative of just spreading the ammonium sulfate. And so either one of those two were pretty good results on their own. The biologicals, by just spraying the biological on the residues that weren't sized with no ammonium sulfate, gave us a very small yield bump. I think our best one on its own was 5 bushels. And we did 3 different biologicals, and that was the best response. And so, definitely, if you were to pick one, the mechanical or the fertilizer was the best response. The biologicals really only performed really well when the other factors were already there.
Okay. And then I know you mentioned before you looked a little bit at the economics, and I think you touched briefly about kind of like the pros and cons of each of those, like with the rollers and how that could be, it's a bigger investment, but it lasts longer. Amongst the three, so you take into account that, that particular situation with chopping, but then if you look at, the ammonium sulfate, then you look at the biologicals, what were kind of the economic effects of those two treatments as well?
Yep. So almost reverse order. The biological input is definitely going to be the cheapest per acre. I mean, we're talking anywhere from $8 to $20 an acre. The ammonium sulfate, then we put out 200 pounds the acre. So that was about 50 pounds of sulfur with 40 pounds of nitrogen. And so that has its cost to it. Obviously, those rates and value of what that fertilizer price is changes year to year. I do know a couple of years ago, ammonium sulfate was the cheapest per pound of nitrogen at one point. It was economical in our study, so we had 8 or 9 bushels, and that did pay for it, but it didn't leave a lot of room for profitability. And then the sizing knife roller, like I said, that's a pretty big investment up front. But then how long do those blades last, right? Depends on how hard you run and how many acres you run them through. Um, but you should get at least 5 years out of those, maybe resharpen them along the way. And so that investment comes over time. So a little bit different perspective there. One of the things I will highlight that I've had good discussions with growers on, fall applied ammonium sulfate, right? Broadcast on the residues. Should we be promoting fall applied nitrogen? That's a good
Hmm.
question, a good challenge to it. And I will say this, the intent was to purposefully use that nitrogen to degrade the residues, which means it would be immobilized by the microbes. As they break down the residue, they immobilize the nitrogen and sulfur in that application. And it was just a 40-pound of nitrogen rate, not a full rate like we think of fall in if we're doing like anhydrous. But it does get one thinking. And we did all this in the fall because one, you've got time, but two, you want to give as much opportunity for that residue to degrade before you go plant into it next spring. But
Hmm.
now we've started doing the next steps. What if we do these practices in the springtime and make sure we hit the environmental water quality? So we're now starting to look at, does spring AMS have the same effect as the fall AMS? And therefore, we get the yield bump with minimizing the potential for that nitrogen not being immobilized and potential leaching over the winter. So every practice and in research, we're small plot, right? We've shown what's potential, but pros and
Hmm.
cons, the economics, and then also just keeping in mind that environmental quality in the back of our minds as well.
Yeah, absolutely. That's really interesting. I'll be interested to see the results then of that that new looking at it a different way, looking at that spring applied treatment. And so, looking a little bit more on kind of the other side of the study at the impacts on the soil microbiome, can you talk specifically about that? I know, over the past few years, the soil microbiome has gotten a lot more, kind of airtime than it did in the past. And so it's really interesting that you guys looked at that as a factor in this study as well. So, what specifically were you looking at in the microbiome? Were you looking at certain species, and then how they changed with these different rotations?
Yep. So this gets exciting. And when you have a focus question, I think there's a lot of value to microbiome work, soil health work. But that's the key there. I would say anyone listening that has interest in this, make sure you have a key question and comparison to make that have value to it. Because if you go to the commercial labs, it's quite expensive for what some of these are charging. So what we have, this site in particular, has replicated blocks of continuous corn or corn soybean rotation. So when I said earlier, we had 17 or 19 years of continuous corn, depending on the site, that was right next to, in replicated blocks, 17 or 19 years of standard corn soybean rotation. And so the first question we had is, after 17 or more years of rotation, what's the microbiome difference between those systems in general? A couple things made sense. We had different elevated fungal diversity under the continuous corn. The fungi go after the hard to degrade residues. Corn stalks are not that easy to degrade compared to soybean stubble. And so that made sense. Higher diversity and slightly higher abundance of fungi under the continuous corn side. But what kind of shocked us, there was almost no difference in the bacterial communities between those rotations. So 17 plus years of continuous corn basically had the same bacterial communities as 17 years of corn soy. That
Hmm.
surprised us. I mean, you could just start to see some separation between them, but it wasn't strong enough to say these are very different. For anyone familiar with the Marrow plots on campus, that's over 100 years of rotation, very different bacterial communities. But what it kind of shows is you need, in certain cases, decades of a management practice to change your microbiome. The idea that you can change your microbiome in one season or in 2 seasons, I would challenge that. And especially based on the data here, 17 years and the bacteria are just starting to differentiate. Why this is important, and going back to the title, implications for residue management. This is just a good reminder for farmers, and they know this, right? Not all residues are the same. Corn
Hmm.
stalks are very different than soybean stubble, which is very different than your cover crop. If you have a legume cover crop, or if you terminate your rye when it's your rye cover crop, when it's a foot tall versus if you let it head out, that
Hmm.
changes all the composition to it. And so the question was, those practices we talked about earlier, do they do the same thing on continuous corn as corn soybean? Because you might have different microbes in the soil. And when I think about residue management, who are you managing or what are you managing? Are you managing the residue or are you managing the microbes in your soils that do the work in the end? And I think that's the
Hmm.
key to the whole thing. That's what got us really excited about it. Um, and I'll just summarize this. All those factors we talked about, chopping, the ammonium sulfate, the biologicals, basically did nothing to have an advantage in our corn soy rotation. And it was really only significantly notable yield response to those practices in the continuous corn side. And that's partly, we think, maybe you needed that fungal difference to get them to respond to the management practices. Whereas in that corn soy rotation, really had no effect. If anything, we lost some yield on a few spots, just a couple bushels. So that's, really exciting. Still trying to tease into, why exactly would it be such a strong dynamic? But that's where, okay, which soil microbes we have probably determines which practice is going to give you the best response, because you're
Hmm.
not treating the residues, you're treating the microbes in your soils.
Right. Yeah, that's kind of fascinating. And, the soil microbiome and thinking about all those fungal and microbial species, I know that would be a whole nother paper in and of itself, right? Kind of looking at what they all do. But did you guys have any ideas of, or many, like maybe speculation of, um, like you said, it takes a long time for these species to change, right? Which to me is kind of interesting because they're microbes. They have such tiny little lifespans that evolution, in my mind, I thought would happen quicker. So that's surprising. But if you had the chance to do this, 50, 100 plus years in the future, do you have any kind of speculation of what you would end up seeing in that, long term of a study regarding the differences in the species?
Yep, I think the trends we saw in fungal would continue to grow. I think the fungal diversity would continue to separate in the continuous corn versus the corn soy. And then I would expect that the bacteria would eventually be different between those. And one of the things I should have started off with, I am an agronomist myself. I am not a microbial ecologist. And so give a huge shout out to Dr. Angela Kent in the Natural Resource Environmental Science Department. She's the collaborator on this project that helped us do all that microbiome work. And she has work on the marrow plots that does show 100 plus years of rotation effects, very clear distinctions. And so it's kind of, this study working with her was great because it's almost like we were able to take a snapshot of what might the marrow plots have looked like at year 20. Versus when she did the study, it was at year 100. So there is some work out there on there that does just show over time it continues to differentiate, but takes time. I think that's the key here. You won't change the microbes in your soil overnight. And it's great to understand which microbes you have, because then you can decide, all right, these are the microbes I have. So these are the ones that I'm probably stuck with for at least a short time, a 5-year window. So let's start managing those microbes rather than trying to change them. And that comes into our third co-author, Dr. Andrew Margonaut, who does a lot of work in soil health and enzyme play. And I would say the work that he was able to provide in this collaboration was the soil function. And that's something I would challenge growers to really start focusing on do you want to change the microbes you have, or do you want to influence the microbes that you have in how they do their different jobs and the function of the community rather than changing the community itself? And that's where we're able to do some of the enzyme work. And Dr. Margonaut identified we had better enzyme potential in continuous corn over corn soy because you have a lot more substrate to work on, a lot more residue to work on. So really bringing it all together. And then, yeah, as I've been rambling the whole time, if I had to summarize, it's, in the short term, and that's 5-year window that I'm saying here, you got the microbes you've got. So focus on managing what they need to then better perform their functions.
Awesome. So I had a couple conceptual questions come up while you were talking. So forgive me if these are coming out of left field, but a couple of things that made me think. First of all, you're talking about kind of managing the microbes that you have. So do you foresee maybe a potential increase going forward of, when folks are doing soil testing, are they going to start maybe testing to see what kind of microbial communities are in their soil? Or is that kind of too outlandish, too expensive? What do you think about that?
It's being done, and there are commercial companies offering, and the samples can be quite expensive. I mean, up to $200 per sample.
Oh,
I mean, if
wow.
you have a 100-acre field and you're doing 2.5-acre grid, I mean, you got 25 samples now that you're going to send in.
Yeah.
I think there can be value there. Now, in research, we're very focused trying to understand, when we have these communities, how do you manage them? Then we'll be able to maybe put that into recommendation practices moving forward. Like I said, this is the type of research I think that's 5 years ahead maybe, but a lot of farmers are doing this. And so what I would say, number one recommendation for those that are doing microbiome or soil health work on their farms, time changes everything. I mean, you can go to the same spot in May, June, July, August, and September, and you will get 5 different microbial community results.
Hmm.
So if you're trying to look at the communities, try and sample same time of year. If you're going to track this in your field over 3 years, always do it at the same time. Don't try and compare, this year I got the sample pulled in May, next year it was pulled in August. That's just going to change everything regardless. So that's my number one recommendation on that. And then also sample before or after a rain and keep it consistent, because
Hmm.
if you sample before a rain and then you get an inch rain and you sample after, different microbes respond much more rapidly to the moisture. So keeping in mind as consistent as we can will give you the best way to interpret your results.
Gotcha. Yeah, that makes a lot of sense. And then my other conceptual question for you was, you talked about how, the different microbes will respond differently to different things. And so that made me think of, with biologicals being kind of that up and coming area, and a lot of folks are kind of leaning towards at least learning more about them, potentially starting to use them. I've heard at least anecdotally, they'll work differently depending on kind of the condition of the soil that you have. And when I say condition of the soil, I think I'm referencing the microbial communities that they have. So, if it was quote unquote good soil, maybe the biological won't react it, or, the crop won't have as much of a reaction to it or a response versus if it was quote unquote poor soil. So, again, looking forward, having an understanding, taking these samples and getting an understanding of those microbial communities, do you think that having that information can kind of give growers kind of some window of insight into whether a biological would be beneficial to apply to that area?
Yep, I think that's where this is going, or where industry would hope to see this going. Pick the right product based on which soil type, or in general, most people presume poor soils are going to respond better than the rich soils. But sometimes we see really good response on the rich soils, because there's a lot more potential. And so,
Mm-hmm.
if you can tap into that potential a little more, it goes a little further. The biggest thing that I would say at this time, your sound agronomic management practices are the best for that system, right? You know which tillage operation is best for each field that you own or rent. You know which fertilizer management strategies are best in play for P, K, nitrogen, sulfur. I mean, you've been farming these fields for years. Your family's probably been
Mm-hmm.
farming them for decades. And so don't skimp on base good management. Where I see biologicals having a potential play is taking good management and making it a little better. Our data in the paper you brought up was the exact thing. The biological by itself really did not do much until we paired it with the sizing and the ammonium sulfate. So the
Mm-hmm.
sizing, the ammonium sulfate got us started, got us going, and the biological was just able to take it one step further. So rather than try to replace a practice, it's a complement to that practice and gets a little more out of it. Some of my best biological results are full rate of fertility with biological gets a little more than full rate of fertility without.
Mm-hmm.
So rather than trying to reduce our inputs, it's just making our full inputs more efficient. And so that's on today's knowledge. 10, 15 years from now, if we revisit this podcast, we might have enough information to say, yep, soil test your soil for the microbiome. Based on that result, we can recommend which microbes you might want to supplement. But
Mm-hmm.
today, we just don't have that background. I don't know of anyone that does. And I would be a little bit wary if somebody claims they truly know. Nope, if that's your microbiome, our bacillus or our fungi are perfect for that spot.
Mm-hmm.
That's a big, that's a tough question to really tackle. So.
Yeah, interesting. So I just have one final question about the paper itself. You mentioned earlier that going forward in the future, you want to look at the effects of spring applications of these treatments. Are there any other different treatments or different factors that you're hoping to look for as it relates to this study for future publications?
Yep. So the timing play, we have a new version out too, is the no-till versus tillage. I'll go back to, if you're interested in continuous corn, we know tillage is still the best thing to minimize the penalty. So what if
Hmm.
you do some of these practices and then work them in? I mean, does it just make that response even greater? So we've got a study out now that is looking at these practices, but with and without the tillage component, along with the timing. Different sources, right? So one of the challenges with the ammonium sulfate, we used a granular. So you have to now have the application costs with that floater going across the field. What about ammonium
Hmm.
thiosulfate, where you can add that to your spray tank as a weed and feed approach, minimize the economics of cost of application because it's going out with the herbicide or the spring nitrogen. There's a cautionary tale there. We've learned this in our own research. ATS burns the vegetation pretty quick. And I mean, if you're doing a Roundup application to kill your cover crop or to kill your winter annuals, and then you crisp up the plant at the top before the Roundup can actually move, you may not get the kill you're looking for because you stop the herbicide from translocating and then the plant continues to grow. But those are the types of things we're looking at, alternative sulfur sources that are maybe more conducive to adding to what you typically do rather than requiring an additional pass across the field.
Right. Interesting. So yeah, that'll all of that will be really interesting to see what you guys come up with. So to switch gears a little bit, moving away from the paper discussion, I want to talk more so about this year's growing season. I know we're in two different areas, but I think throughout the state there has been kind of consistent weather patterns in terms of we've had a lot of wetness this spring, which I know reflecting on last year is very relieving. But I wanted to know, how is the growing season going for you guys? And with those early season conditions, kind of what have you been seeing in fields as you're doing your work?
Yep, I think everyone's dealing with the same thing of there's pockets of go time and pockets of downtime.
Right.
And some places have been luckier than others. I know Western Illinois is pretty far ahead, if not almost done. I know Northern Illinois actually had quite a few windows and a good amount of acres up there, pretty well done. Then you get into Central Illinois, East Central, and then Southern Illinois. There's pockets where there's still, half the ground hasn't been able to be planted yet. One of the things that we are seeing, there was that April window, we prioritized soybean over corn. And I am really pleased with my soybean stands. They came through, they look good, strong. And I know a lot of the April corn in some areas sat a little bit or it got a little cool or it held some water and there's uneven emergence. So one of the things I am seeing is if you planted in April, it seems like the soybean tolerated those conditions a little better in terms of final uniformity, whereas the corn, there's a lot of emergence variation in certain areas for the April corn. I've also seen some people side dressing already, some cornfields that look beautiful. So
Mm-hmm.
it is very pocket dependent. Biggest thing is, don't let what your neighbors are doing affect you. You know your fields, you know what conditions are best for optimal planting. And so just do it as needed. It's the 28th today. I know we're starting to come up, especially in Southern Illinois, on some of those prevent plant dates and making those decisions. At what time do you stop, put the planter away and take that prevent plant? I'm good putting full maturity corn hybrids in through May, which means I got 3 more days before I start deciding if I need to lower my maturity group
Mm-hmm.
on that side, or farmers are making the decisions, at what point do we keep planting, but just switch our corn acres to bean acres. Biggest thing there is to remind you, what did you already spray? What did you spray last year? Make sure that you're
Yep.
not going to have any plant back issues if you do change crops. But a lot of variability, but, I think the progress this last week was probably quite good. We put in, I think, 30 trials last week. We
Jeez.
had a good window last week, so I expect the next progress report to be quite good for Illinois later end of May, but I think we'll be less in that June window than we thought we might be.
Yeah, absolutely. So looking forward then, like you said, we only have a few more days or a couple more days really until we hit June, which is insane in itself. But looking forward at June, what would you say growers need to be on the lookout for as it pertains to, what conditions might kind of change maybe some different management aspects for the rest of the growing season, at least the early portion of it?
Yep. So the number one question that farmers will be asking is about side dress nitrogen, or if you're an all pre-plant nitrogen system, and now with all the rains we've had, do you need to put more on or not? There's a couple of conditions there because sometimes if you have a reduced yield potential, more nitrogen does not guarantee a response because if your yield ceiling has been capped based on certain conditions, it may not need that extra N. But there are certain scenarios where certainly a little bit of nitrogen might help. So keep an eye on it. One of the things to remind you, though, if you are going to put on extra N or when you do your side dress plans, if your soils are still wet and holding water, you need good aeration and good soil conditions for roots to be active. So making
Hmm.
sure that we do time that nitrogen at a point that the crop can use it. If we just pour it on now and our soils are still, dry on top, but really wet in the root zone, the roots may not be taking it up. So those are things to consider. But one of the things that our research has really showed us the last 2 years, if you have a dry June, that really limits the total yield potential. In the last 2 seasons for us, 22 and 23, we had very minimal response to management in season. That would be, higher densities, the narrow rows, side dress nitrogen, and foliar protection being fungicide. they were really close to breaking even, but not profiting necessarily. And we really looked at the data and looking at our kernel counts, and you determine ear size. This is more corn than soybean focused discussion in
Mhm.
those early vegetative phases. I mean, but by V12, the potential of that ear is already decided. After V12,
Right.
it's protecting it. And if you have a dry June, you're going to have smaller ear potential. And that's where maybe the extra management is. You can do it, but if it has nowhere to go because the ceiling's too low, you may not see the response. But if we have good precipitation through June, even if it's late planted corn, I mean, if your corn went in after May 20th, but we got good moisture in June, you will still have high yield potential. And so don't give up on management at that point as well. So, where I kind of stand today is keep to the plan. Don't throw those plans out the window yet for what you had, especially if we get good precipitation in June. You'll have higher yields than I think you might expect at the end of the year, given a later planting. If June turns off really, really dry, maybe then we start making decisions on holding those late season foliar applications or minimizing how much side dress N we may put out if the crop's not going to be able to use it.
Mhm. Interesting. Yeah, I think that's really good information for growers to know. I know with last year there was a lot of those questions of do we do it? Do we not do it? Is it going to do anything? So that's great that you guys have data and information on whether or not that's actually, prudent to be doing in those different conditions. So switching gears a little bit here, I know something you and I discussed kind of offline, was talking a little bit about soil health testing, and this kind of resonates with the paper that we were discussing earlier. And, obviously soil microbes are part of soil health. And so soil health testing is something that's being talked about more and more. And it's just one of those things where, is it something to be done? Is it worth it? Does it tell you anything really? Kind of what are your thoughts on soil health testing? And can you give some information just briefly about what is being tested for in a soil health test?
Yep. So the more common ones, like many people look at the Haney test and things like that. But there's three big areas that I would say are newer that you can get, an evaluation on. So you send a soil test in, and they'll give you the P level, the K level, the nitrogen level, right? So what else can we add to that list? The big one that I'm seeing is respiration. So that CO2 burst, where they say, how active is your soil based on how much carbon dioxide it gives off when they run that test. So respiration test is one. The other one is soil enzyme testing, largely on carbon, nitrogen, and phosphorus enzymes, a little bit of sulfur as well. So that goes back to my comment about that's soil function.
Hmm.
So microbes produce enzymes to do the jobs of residues decycling and mineralization and mobilization. All those processes occur with enzymes that the microbes produce. And so when you're measuring soil enzymes, you're really getting an idea of what's the function of my soil. And then now there are companies that will actually give you basically a DNA test of your soil. Um, there's really fine tuning where they can tell you every single individual you have out there. That's a lot to work through and hard to interpret. So what I've seen now is reports that are on a scale of 1 to 10. You have a high pathogen load or a low pathogen load, or your beneficial load of bacteria is less, but your beneficial fungi are really high. One of the challenges I have is who sets those thresholds, right? What determines high beneficial and high pathogen? And is that threshold the same for my 2% organic matter soil in Southern Illinois or my 6% that I work on up in Northern Illinois, mollisol or alpha
Hmm.
sol, right? Probably not. So those thresholds and stuff, that's my cautionary tale. When you're doing those DNA testing reports for your soils is how are they deciding their benchmarks? What determines if that is adequate or deficient for a certain class of microbes? And then the newest one I'm seeing is these fungal to bacterial ratios, right? Your microbial biomass, what portion of the microbe biomass is fungal versus bacterial? And I
Hmm.
think there's this general rule of thumb. You want it, and don't quote me on this, like a 3 to 1. For every 3 units of fungal biomass, you want 1 unit bacterial biomass. But again, what's the threshold? Should it be 3 to 1, 2 to 1, 4 to 1? And so these are the things that are being offered. But I my biggest caution is who's deciding the thresholds and where's that justification. That's what I think is missing in a lot of cases. Now, I love the idea, though, if you have a focused question. And this is in our work. We have replicated small plot. So I sampled them on the same day at the same time, and I'm comparing management A to management B. This is where I think if you're a farmer and you're interested in doing this type of work on your farm, have your comparison points, right? Maybe you're keeping one field a strip till, but taking the other to no till, right? Sample them on the same day, compare them direct to each other so that those soils get the same treatment the day you pull them out and send them to the lab. If you're tracking year over year, target similar time of year, always in May or always in April or always in the fall, rather than having a month split this year's sample to next year's sample. So that's, I think, the biggest thing moving forward with this. If you're interested in it, because I think there will be potential, and we will eventually have recommendations to pair with these, be consistent in where you sample, when you sample, and what you're comparing.
Yeah, absolutely. It's really important to understand, what's being tested. And I think those thresholds that you talked about, having a base point to go off of is really important because otherwise it's, what is that information anyway if you don't have that base point to be cognizant of. So yeah, I think those are really good points. So we're kind of wrapping up now. So I have a couple, or really just one kind of fun question for you. Do you have any research updates that you want to share, or are there any certain projects or trials that you're currently working on that you're, particularly excited about? Anything you want to share in that realm?
Yep. And actually, this will be the opportunity to jump onto soybean. We've been talking a lot of corn.
Mm-hmm.
We've got two projects on soybean. One is a biological one, but it's supported by the United Soybean Board. So really excited about this. It's 10 commercially available biological products, but we get to look at it entirely independently. It'll be a public report for everyone to see. We've got it in Washington, Champaign, and Kendall counties. My Washington County is not planted yet, but my Champaign and Kendall County are in the ground. So a good diverse set of locations there to look at different types of biologicals. It's not a product focused question, it's a type. So we have 10
Mm-hmm.
products that represent 5 categories. So traditional inoculum, fungi, a brassica mycorrhizal fungi, bacillus, non-traditional end fixing, and then just a non-traditional microbe package. So that'll be a fun one to work with United Soybean Board on. And then the Illinois Soybean Association, we have a collaboration that we're working with them on tissue testing. When we think about tissue testing for soybean, what do? We sample the most recently mature trifoliate because that's where your most active growth was. And if you are deficient, it should show up in the most recent vegetation to grow.
Mm-hmm.
But when we think about soybean, right, we're still putting nodes at the top of the plant, but at July, you'll have flowers at the top and pods at the bottom. Do you
Mm-hmm.
really think the top of the plant is doing the same thing as the bottom of the plant in terms of nutrient remobilization? And it's probably not. So we're going to sample every trifoliate at every 2 growth stages to try and determine when does the soybean start moving nitrogen or other nutrients from the leaves to those developing pods. And pretty sure we're going to find out that remobilization starts earlier than you may think in the lower canopy, not as soon in the upper canopy. And then we'll be able to use that to maybe make better foliar micronutrient application recommendations. So really excited about that work. It's kind of just another look at tissue testing, whole plant level, not just top of the plant, and assuming that's going to be adequate for the whole plant system.
Wow, yeah, that sounds super interesting. I'll be really interested to see to read that when it comes out. So yeah, that's really great. And so the last thing I want to mention before we wrap up, way at the beginning of our conversation, I talked about how I always learn so much by listening to your group and the presentations you give and talking about all the research that you guys do. And where I did this last year was at your Crop Physiology Field Day. And so, are you guys doing that again this year? And do you have any more information that you'd want to share with the audience about kind of the when, where, who, what, why, all of that?
Yes, yep, and we do. So the 3rd annual Crop Physiology Day with the theme this year of Got Yield is on August 1st. That's a Thursday. And that's here on campus, 4202 South First Street, for those wanting the exact address. We do start early, right? 8 a.m. to noon with different presentations from the grad students, myself and Dr. Bilo. We'll have our equipment out there so anyone can ask, when I give you data or when we talk about a paper, how was that developed? Do you trust the equipment we used and how it was put out there? So all of that's on display for Q&A. Great topics this year on fertility, yield components, gosh, off the top of my head, residue management. I mean, we're basically going to touch on everything that we touch on in our research program. So August 1st, 8 to noon, and then lunch. So free food, free everything. And we'll have a registration link here and start floating that flyer around pretty soon. But yes, August 1st, 2024 on campus.
Great. Yep. I hope I can make that. I'm gonna try my hardest too. Again, like I said, super great event. I learned so much last year. The food is also phenomenal, and like you said, it's free. So I'll plug that for you too. So, Connor, thank you again so much for being here today and talking with me. And I wish you the best of luck for the rest of your guys' growing season.
Thank you so much. Always appreciate the opportunity to talk, help with extension, get the word out there, and for highlighting that paper. So we're really excited about that, and all the other projects. So stay tuned, and I'm on Twitter, LinkedIn, and we have a website. So we'll be posting updates all year long.
Perfect. Thanks, Connor.